I wanted to tell you a story. It's it's a story that comes in stages, but fundamentally, I want to talk with you about an issue that I think we all know is important and still somewhat mysterious. And that is how we can go from making a decision to do something and then actually doing it. To me, it's a miracle. And the thing that we do know here is that motivation is absolutely crucial to this whole circuit of decision and action. We rely on this and we're really devastated without emotion and motivation to help us. But if we can complete this change, then what we know is that we can affect movement and thought. And it's really been an extraordinary thing that in over time and quite even up to right now, it's still a surprise that it turns out that the basal ganglia is so deep hidden in the brain, so deep underneath the beautiful neocortex, could possibly be so important as they are in this. And I'm showing them here as just a little pink tip of the iceberg regions in a very famous, uh, one of the early scans, levodopa scans of this region. We also know that if this circuitry is broken, then what can occur, what will occur, are extrapyramidal motor disorders, for example, Parkinson's and Huntington's disease, dystonia of various sorts, but also a very large range of neuropsychiatric disorders. They go all the way from disorders that are psychomotor, like OCD, Tourette, to anxiety and depression, and then to our own kind of uh, so-called normal behaviors, learning, then excessive uh, learning to the point of addiction, the effects of stress. And I want to show you here at the start a kind of cartoon of the basal ganglia system, which is a very, very old one. I showed down here at the bottom, it's thought to be at least over 550, 560 uh, million years in age and maybe even older. My own interest now is in following the system in Drosophila of all things. It's ancient. But the major thing you can see here is from the neocortex, great pathways that go down to the center of the basal ganglia, the striatum, the main input-output channel, then out through the output centers back to the neocortex. And it's thought that through these looping pathways, which are modulated by reinforcement signals from the dopamine-containing neurons in the midbrain, that through these circuits, we can help select what to do next, or what to do longer term. These are the famous uh, 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 release inhibit pathways of the basal ganglia, very well known in the motor field. But there are other pathways that I want to talk to you today about one set of circuits, one circuit in particular that leads also from the neocortex, but instead of going directly into these loops, runs through the striatum in specialized zones, which I'll come to, which lead down directly to the dopamine-containing neurons of the substantia nigra. This is a great privilege. Most of the neurons that project down directly to the dopamine-containing neurons come from this circuit. And the circuit has indirect here through the habenula, down into that same circuitry into the serotonin system. And what I need to do to start out this story is to tell you first about a very old finding. I have to reach way on back because it's begun to pop up a lot now in what we do. In fact, we've become very, very re-interested in this uh, old finding. What you're looking at here is a cross section through the striatum of a human uh, brain. It's been stained in an old-fashioned way to show the cholinergic system of the striatum, and that's shown in the kind of darker hue. Here's the internal capsule caudate nucleus putamen, and what you can see is these funny little pale zones that seem to have less evident expression of cholinergic function. We came to call them striosomes for striatal bodies, uh, and found, along with many, many other people in the field as time went by, that these are extremely highly uh, differentiated in terms of their neurotransmitter related characteristics, their inputs and outputs. They are, in fact, these zones here that I showed in blue that are in a circuit that leads from regions of the cortex related to evaluation down to the dopamine containing neurons of the striatum. This finding stayed at the level of more and more neurochemical, neuroanatomy kinds of work until we got a break. And that break came when Frank Eblin, then a postdoc, and I began to ask what parts of the cortex project down to the striosomes. And what we found was shown here in a cartoon of various old-fashioned tracer injection sites in the medial wall, in this case of a macaque monkey brain. And it was the blue zones just in front of the genome of the corpus callosum, just this little blue region that we found differentially projecting to striosomes, along with a companion in the very caudal part of the orbital frontal cortex. This became extremely interesting because soon, as human brain imaging became possible and more sophisticated, it became known that that same pre-genual region was abnormal in depressed people. Here you can see from Nora Volkow's work lit up in cocaine users who are in a craving stage. And here, in a fascinating study by a German group, this region was differentially activated in migrants who came into Germany right after the border was open, first time in urban environment, and suffered, therefore, great anxiety. So when we started, we had no real appreciation of how extensive this system was. But I can show you now, with the help of Kang-Hoon Chen and his wonderful SHIELD method, the extent of it in a mouse brain, which we've engineered so that the striosomal neurons are red. And as I run through this, this is a nose up image, so the frontal lobe is up. If I go through this without making another error, you'll see in the video a zoom into the striatum. And now look as we go through how these clumps, clusters of cells form, and then they form a kind of labyrinth, weaving all through that system. It's really, this is a very, very large set of neurons forming that circuit. I'm having trouble with advance. So the question was, if this is there and this system is so extensive, can we possibly figure out how to tackle it in a behavioral way? and in a way in physiology. And it was at this point that we had uh, the idea to go after decision making. Because if this system really was part of an evaluation system that could lead down to the dopamine containing neurons, surely it should be important. And it was evolutionary old. So when Ken Amamori came to the lab, we decided we'd look at something called an approach avoidance conflict decision making task. And we did that explicitly because in human clinical work, it had been known for quite a long time that if humans are faced with a conflict between having to accept or reject uh, an offer made to them, but the offer contained good and bad, so they had to take the bad to get the good and vice versa, that this kind of decision making was very vulnerable in people who suffered from anxiety and depression. You can actually see uh, an example of this here, of its conservation. He is looking right at you, or anyway, right almost at the camera, deciding whether to approach or avoid. And his friends, can you see them in the back? They've already long since decided to run. They're running away, they're avoiding. And so, again, for that reason, it's evolutionary conservation and the fact that the system of decision making is so vulnerable in people with problems, with depression and anxiety. Ken and I set up this task for macaque monkeys. And so we could then uh, have them look at compound offers, good and bad combined offers, and we could plot how much cost, we could plot how much benefit, we could get a decision boundary here. And you're seeing here actually a reduced plot of the uh, uh, approach and avoidance decisions made by one. And the beauty of this is that we can get numbers. That's what we really wanted. So we could quantify when we try to manipulate the brain, we could quantify whether we had made a difference in the approach or avoidance behavior. Had we shifted that decision boundary? And in fact, we did. Here you can see a little schema showing uh, where we micro stimulated in that same pregenual region. We did that because through very extensive recordings, Ken had found that there were approach favoring and avoid favoring neurons down here all mixed up together. And when we microstimulated that zone, what happened was that the uh, avoidance was increased. It was increased solidly in a small handful of very precisely located cases in that blue zone, as though the animals were overvaluing the outcome as in terms of its negativity. So we tried to see whether we could block that behavior using diazepam, an anxiolytic used in humans, that that did block the increased avoidance dose dependently. And we always carried along an approach task, so no real conflict, just larger or smaller reward. And there was no effect of the microstimulation whatsoever. So we wondered then, and we still are wondering increasingly actually, whether what we were seeing were proxy states for pessimistic decision making. Were we seeing something really related to human uh, anxiety, depression, or some mix thereof? And so we've begun a series of experiments in collaboration with Diego Pizzagalli at McLean Hospital here in Boston. And what we've uh, done so far is one study. We're the second study will start on Saturday, actually. It's coming Saturday. And I'm showing you an example of what we found. What's special about this study is that we actually had the humans look at precisely the same kinds of stimuli we used in monkeys, these horizontal red and green bars, red for reward, yellow for in for monkeys it was air puff to the face, for humans it was unpleasant photographs. And you're seeing here the increased activity in that by now familiar pre-general region in subjects who avoided, but also who had the subjective impression that the offers were aversive. So this is our first real solid hint that maybe what we were doing in the monkey had significance for the human. And I can tell you now that other groups are actually several groups now working on approach avoidance using basically our paradigm. And so far the results are similar and augmentative to our own. But we knew we had to go to a cellular level, a circuit level mechanism level. And so what we did was to try to generate mice in which we could do a better job of knowing whether we're recording or seeing striosomes or matrix. And here's what we relied on. I found many years ago that striosomes are born, quote, before matrix neurons. And with Sean Kelly in Josh Huang's lab, we were able to introduce tamoxifen burst aiding method, which we did. And I'm showing you now the evidence, just briefly, that it is apical progenitor cells in the very early germinal epithelium that are destined to become primarily striosomes. You can see the red here. This is now a 28 postnatal day animal whose tamoxifen burst aiding occurred very early during the burst aid period for the striosomes, E10. And here, what you can see is that, in fact, it's not apical, but basal progenitor cells that produce the matrix. You can see a comparable 28-day-old animal injected now at E17.5. So to our great interest, this distinction between the future striosome and matrix compartments is already present in the germinal epithelium, and conserve, which we have used as a permanent marker for the compartments. It's not perfect, but it's not bad, so that we can do now behavioral experiments and other kinds of experiments with some knowledge of what kinds of cells we're doing. So we produce lines of mice, and we're perfecting them all the time. We started out with Alexander Friedman and several others and wonderful people in the lab to set up an approach avoidance or cost benefit task for first rats and then mice. And simple T-maze, we've used a lot of T-mazes in our lab in study of habit formation, but this one's special because the animal on one end could get pure chocolate, I'm pointing to it here, but with that came bright light. But on the other side, he had dim light, so didn't mind at all to run that way. But the chocolate was very diluted, and we actually formed dilution curves, and we measured the lux and did psychometric studies for every single animal we ran. And we found, as you might suggest, that most of the time, mice and rats run to the dilute chocolate side because they want to avoid the light. It's important that I mention that we ran other decision-making tasks, like in the monkeys, we had a benefit, but we also had cost, with reverse cost benefit. So dilute, strong chocolate goes with low light. And the reason this is so important is that most of what I'm going to talk about now occurred only in the cost approach or approach avoidance version of this task. So let me show that to you. And what we did was we had tetrodes at first, later photometry, recordings in the prefrontal cortex region, PL projecting to this, mainly to the striatum. In the first days, we had to use the hard to do ortho-antidromic identification. Later on, we had the engineered mice. And what we found was that when animals are behaving normally, in fact, what's going on is that the prefrontal cortex projecting down to striasomes, that region is active, but the striasomes, to our amazement, were off. So then we said, well, why don't we just damp down that cortex and see what happened? And you can see here, if I blink back and forth, instead of running to the low light level, they ran to the aversive side by an increase of 20-25%. And in the circuitry, what we found was that now, the cortex was almost off because of us. The inhibitory interneurons were turned off, and striasomes now were blazing. So what we found in short was that what we had to do was to slow down what normally is a very fast inhibition to make that circuit work. So we found, in a way, my sorrow is that what do striasomes do? If the striasomes are on, the mice approach the versus stuff like light. Thank you. And striasomes are off, then the mice can behave normally. But this actually raised a question, which is, what on earth have we done by manipulating that circuit? How can you make an animal flip his decisions that way? And so could the striasomes actually be in some way a switch that has to do with changing state? Well, we're the lab we are, and we are, how will we do a state change in some feasible way for our lab? So we decided we would go immediately to find out whether experience alone could do that. And so we decided to use mild chronic stress. 2 weeks, 1 hour a day, mild, put constraint, put the little mouse in a cloth bag, hour a day, 2 weeks. David brought up in his introduction about a 6-hour flight on an airplane. And I want to say that 6 hours in the middle seat is about what I think the animals were experiencing. We also did mild shock because we had to do a second thing. And here's what we found. Here's the no stress baseline. They're running what we call rationally to avoid the bright light. Now, the striasomes are off. Now, these animals have been stressed. Nothing else has been done to them. The calvarium has not been opened. They start running to the bright light side. In fact, to tell you the truth, we couldn't tell the difference between the behavioral curves of these mice and mice in which we manipulated the circuit, which gave us the thought, one, experience alone can switch the circuit dynamics amazingly. And second, could we then, if that's related to what we saw with the circuit change, could we reverse the effects? And so we tried to do that. And the way we tried to do it was to go down to those inhibitory interneurons. And when, in fact, we turned them on, we completely blocked the effects of stress. It was as though the animals never had been stressed, and we found this in mouse after mouse after mouse, really tens and tens of mice. What this kind of experiment was telling us, first of all, I had mentioned that it's a matter of dynamics. It's who gets there first. Who gets to the striosomes first? Is it that prefrontal PFCPL region, or is it those little interneurons which also receive input from the neocortex? Is it neocortex? Is it local circuit? The dynamics are critical. And if you can restore abnormal dynamics, you're back in business in terms of dealing with chronic stress. But it also makes another point, which is that this circuit is incredibly vulnerable to even tiny, tiny little changes produced by either the external world or endogenously by some internal state change. So that led us to the next question, which is that if some kind of environmental effect can be so profound, shouldn't learning somehow be involved in this? That's a pretty strong state changer. And so we set up yet another task, and I'm this time talking about experiments that were real recently published by Alexander Friedman and Emily Hoekstra, who is a co-first author. I wish they could have their names sort of overlaid, because Emily Hoekstra is extremely important in this work in many, many ways. But here we changed to a task in which we had two tones. One tone came on, and it could be 1, 2, 3 amount of tone. The more tone, the more licks, the more reward. But for tone 2, which was going to indicate a bright light coming on, and these were head-fixed animals now not running in a maze, the fewer licks, the less cost. So this is called a valence discrimination task, as many of you will know. And we found it very, very useful. First of all, we could take measurements of how well the mice were performing the discrimination. We had an index for that. And then we could look at the activity of the striosomes in matrix, which I'll show you in a minute. And the way we are going to show this is that we had, in this case, photometric recordings, and we could subtract the cost levels of activity from the reward levels of activity to get a response period value, where response period is just as soon as they were able to respond. And you can see that there was a very clear correlation. Each dot is a different mouse, 8 mice, clear positive correlation between the striosomal activity and learning of the discrimination. But when we looked at the lick rates, we found no evident correlation at all. So this is very, very striking. But what was even more striking was when we did the same kind of calculation for the matrix. Here you can see also for the discrimination learning, this time there was no particular correlation at all between the activity in the matrix and discrimination ability, but a very strong correlation with the licking activity. We found this again in a smaller number of animals. We actually since have confirmed it in more. So we knew then that there probably is something quite special about the striosomes in some form of value-based discrimination learning. And we also know that great controversy about learning and how well we learn and remember as we get older. So we decided that it would be a good idea in this sort of experiment to try to look at mice of different ages. And so what we did was to start with what we thought might be the equivalent of about a 35-year-old person, healthy, can remember anything, learn anything. And then we all know what happens with age. And so what we chose was to study mice beginning around 6 months. These are rough equivalents, 9 months, and then 12 and a lot more months of age. Same task. And here you can see the first result. First, let me say that the y-axis shows you the percent of mice that learned versus the young, middle, and old groups. And I was, of course, personally quite pleased to see that even the old guys learned quite nicely, thank you. I will admit, and maybe mention in a minute, it did take them a wee bit longer, where we could be very large amount longer. But what we found that was not so pleasant and to us greatly interesting was that when the task learning began, when the acquisition phases were early, the older animals, even the middle-aged animals, really had a deficit in engagement. That is to say, doing it, wanting to do the task, actively being in the task, until finally there was quite a large deficit, although eventually, given enough time, the old guys could learn. And so what we asked was whether there could be any relation between that decline and the striasomal activity. This ellipse shows the early stage the late stage for the animals, and we're going to look also at the early ones. And so here you can find now for the young and middle groups separated out the striasomal signal, as I've shown you before, versus engagement level. And it was really quite clear that there was a high correlation between the amount of the ability to engage, especially during the early times, the ability to engage and the amount of striasomal signal. So engagement declines across learning, and striasomal activity is correlated with the amount of behavioral engagement that we can detect, at least in this task. So we again asked if there is this going on, could we possibly change that? What we really want to do is learn how to increase engagement in older mice, to say nothing of humans. And the way to do it, we decided, was to try using human genetics. So I'm showing you here a plot. On the x-axis, you see the change in striasomal signal level. This is by photometry we measured, as a function of the change in engagement on the y-axis. And the colors showed you that we did CNO dose response manipulations to find the best doses, but also just to make sure that we could see this is dose dependent, which we did. So here you can see what happens when we increase, when we excite the striasomes. And here you can see when we used an inhibitory dread in another group of animals, how we changed the signal in the opposite way. So engagement levels can be elevated by increasing the activity of striasomes. It doesn't happen, it's the reverse actually, by inhibiting them. And so what we now are wondering is whether this so-called engagement really is a proxy for motivation, for kind of get up and go. And so here's how we've looked at that. First of all, First of all, we have looked at the number of putative parvalbumin-containing contacts on spiny neurons with very high resolution microscopy, which I thought I could show you, and now I am. And we found that in animals, there are some animals that just don't learn. And in those animals, there were far fewer of these contacts. This is highly replicatable, and you can see some of those contacts here in a very high resolution image of a medium spiny neuron in the striosome from an animal who learned. Why could this happen? The first obvious possibility is going back to what I told you earlier at the beginning, which is that striosomes have a privilege, and the privilege being that they can project directly to the dopamine-containing neurons of the striatum. And I want to show you that in what we found in a spectacular image that we didn't realize we would find. You're looking here at the substantia nigra in a cross-section through the brainstem of a mouse. The purple pink is showing you the dopamine-containing neurons. You can see here the white and also green. What you're seeing actually is that the striosomal axons that have been labeled by us, by engineering, are crawling up bundles of entwined dendrites of dopamine-containing neurons that are in clusters. You can see one here in the nigra. And if I show you this, it's one of my favorite images. This is obviously a bouquet of roses, the red roses. Here's the stem. But really the red roses are the dopamine-containing neurons. And the stem is showing you the intertwined descending dendrites of those dopamine-containing neurons. They're bundled together. We call these bundles dendrons. And they're bundled together, and entwined with them are the striosomal axons. And all of this is encrusted with pre and postsynaptic markers. So it turns out I can't show you too many images or I'll run out of time, but it's an extremely powerful anatomic image, an exuberant possibility for control of the dopamine-containing neurons by striosomes. And what that led us to think is maybe if the dynamics of the striosomes are so important, as I said, for controlling state, mood, maybe they could turn on and off sets of dopamine-containing dendrites. And we published that paper, Jill Crittenden and myself. I'm reading the date myself in 2016, and really within a couple of weeks, we were on the phone talking with first one, then another lab. Slice studies have come out. Here's McGregor et al., the Evans paper, beautiful papers, and there are a couple of others showing that in slice, if identified striosomal fibers innervating striosome dendron bouquets, if those are stimulated, then the dopamine-containing neuron activity can be shut off cold. And it also can happen that after this strong inhibition, there can be strong rebound excitation due to the expression of different channel types. So we're wondering now, and trying hard to get at this, can the dynamics of the striosomal cells themselves under the influence of the cortex turn on and off these neurons? It looks like that's true, and this might be a circuit for changing mood, motivation, propensity or impulse to move itself, work on decision-making, work on the ability to learn. So we know that if we want to move somehow, as I personally so much want to do, to move this toward translation, with the aside that I think we all, I hope, have the firmest faith and belief and cherish pure science for its own sake. But how wonderful if we can do pure science and do good at the same time. I'm sorry, that's my uh, goal in life. Um, how do we go on that path toward translation? So we started a collaboration with Miriam Heyman and Manalis Kellis, others at the Broad Institute, and going after what the selectivity of gene expression in striosomes and matrix. We started with a template from earlier work from the Heyman lab, in which they had been able to look at D1 or direct pathway movement promoting cells from the striatum. And then they're down here, an indirect pathway neurons movement, so-called inhibiting, or release inhibit neurons. So, and we are showing you here uh, indirect pathway SPN. It's not actually a Tisney plot, it's an action plot kind of dimensionality reduction I'm showing you here for the matrix indirect pathway SPNs. And you can see here matrix in green, here striosomes in purple. So there's subclustering within the indirect pathway set, and here for the direct pathway set, again, subclustering into matrix and striosome neurons. And these are findings in normal human, and here are findings in mice from us. And you can see, I don't have them quite at the same magnification, but you can see very similar plots in the mouse and the human. And we're very, very excited about this because we're now interested in trying to use this sort of information. We're doing a lot of analysis, as you can imagine, use this information as in a way whereby we could maybe test for vulnerabilities of different kinds of cells. And we're doing that now first for Huntington's disease. But showing you images of striosomes and matrix reminds me to tell you that I've completely neglected the gigantic matrix which lies around striosomes. And we are very interested in working on that. And I just want to show you what still is our rough impression of what might be going on, that there is a learning architecture embedded in the striatum whereby the neocortex can send out information streams to local experts in the striatum, which can be influenced by local striosomes before the network can then bring in modifiers such as dopamine, serotonin, a number of other neuroactive substances, and then select. And here is a cartoon of that. It's actually a cartoon of our modest little theory that the striosomes, which are sprinkled through the striatum, as I showed you in the clarity video, actually the matrix also is modular. You just don't see them neurochemically, but there are modules there that we've worked on, and just a few others, and we're hope to go back to that study. So depending on the type of activity that's going on in any given set of matrix modules, the striosomes can affect one or another behavior. So what I may have been showing you is really selective for a very important prefrontal PACC orbital frontal to striosome system that can help to control mood, motivation, affect, drive. And I want to end then by saying it certainly is inspiration to do the RNA-seq and much more, given what we already are learning from our even immunohistochemistry and RNA-scope, seeing that this system is encrusted with opioid receptors that was known a long time ago, serotonin receptors of certain subtypes that are targeted in psychiatry for psychotic disorders, and here's CB1 receptors, the receptor, for example, for marijuana. So I do want to end by thanking you again. What an honor to be here. I want to end again by coming back, as I did, to the disorders that so many of us suffer. And I want to thank the wonderful people in our lab, starting with Ken and his wife, Satoko Alexander, here's Emily Hoeksta, their collaborators, Bernard Bloom, and has just completed a study using a bandit test, which we're writing up. And here Ayano Matsushima and Joe Crittenden, and the funding for which we are really eternally grateful. Thank you very much.